So yes, this is just an extra video because there were some unclear points in what I was explaining, and also the quizzes made me realize that maybe some things are not so clear. So some of you pointed here that there was some unclear points about the relationship between effective population size and a factor like sex ratio. So just to summarize what the effective population size is. So this is the size of an ideal population with no problem, let's say, that would give rise to the same genetic features such as genetic diversity or number of alleles as the population that you are observing. And then there was this relationship between sex ratio and effective population size, which was negative, like the more biased sex ratio, the lower effective population size. So if we go back to this, so let's take the case of a selfer. So in Arabidopsis thaliana, most of you know it, it's a plant which is able to self, so having sexual reproduction with itself. So imagine that you have one individual here that will self, leading to 99 progeny, so for a total population size of 100. So my question here is for you is, what is the effective population size? So just pause the video, think for yourself, and then I will give you the answer, and let's continue. Okay, so once you thought about it, maybe you found this, and the effective population size in this case is 1, simply because these 99 individuals are basically clones, which means only one individual is enough to capture the whole genetic diversity of this 100 individual population. Okay, so like one parent gives rise to 99 progeny, but because they are all the same, there is only the need of one individual to know all the alleles present in this other 99 because they're all the same. Now let's take a different case, so pangolin, to match the current situation. So imagine this time that you have 50 unrelated, completely unrelated individuals, genetically I mean. And each of them then, whatever, male 1 with female 1 will mate, and male 2 with female 2, etc. until number 50. They will give rise to 50 individuals that are the progeny, so total, again, they will form a population of 100, just like in this case of thaliana. Now the question is for you, again, what is the effective population size of these guys? So again, pause the video, think about it, and once you think of an answer, just continue to play the video. So yeah, so the answer here is 50. And why 50? Because each of them here are unique. To know and to kind of capture the whole diversity again of this population, you will need at minimum 50 individuals, these 50 original individuals. These ones will not increase the effective population size. Why? Because they inherit alleles from each of their parents, respectively, which means they are not unique in that sense. So in that sense, only these 50 unique parents to start with will tell us, okay, this total population of 100 has actually effective population of 50. And we only need to know 50 individuals to capture the whole genetic diversity of this population. Okay, so if we go back to this case. Now, so imagine, like before, like for the pangolin, we have basically a balanced sex ratio. So there is as many males as females. So it's a case here. And imagine that in this case we have an effective population size which is 100, which means that basically each of these individuals are unique and each of them will mate. And if everything goes fine, if there is no drift, if there is no change in allele frequencies, again, because of selection, because of whatever, you can imagine that generation after generations, this population will remain as effective population size of 100. Okay, 100 because, again, the original number of unique individuals is 100, and again, if there is no problem of, let's say, of selection or whatever, then this effective population size will remain the same. Okay, now let's take the opposite. Let's take the case of this one here. So this is, we have 1 male for 99 female. Okay, so of course in both cases we are talking about 100 individuals in total, like population size. But here, so if you have a distorted sex ratio, of course the first generation will still possibly have effective population size of 100 if each single female is completely unique, okay, and also different from the male here. But things get a bit more complicated when you go to the next generation. If you go to the next generation here, every single individual are actually related because they all have the same father. And because of that, then the population, the effective population size will strongly decrease. And especially why? Because generation after generation, you will actually end up in a very similar scenario as selfing. You will actually increase the rate of homozygosity to start with, and especially the rate of homozygosity of the male genome. And because of that, generation after generation, the population's effective population size will simply decrease to reach something close to 0 here. And why again? Because we are approaching something which is very close to the selfing situation, because all the individuals are more and more related generations after generation, because one of the parents, so the male in this case, gave rise to the whole generation to protein here. So I hope this is clearer. Again, if this is not clearer, let me know and I will find another way to explain it. Okay, so now about the quiz. So there was this question about Taji-Mazdy, and what I didn't really understand is quite some of you answered wrongly, but then you all got the right answers in the next question about Taji-Mazdy, which was already a bit more advanced. So maybe it was just the way that the question was asked. So just to make sure, Taji-Mazdy makes a difference. It makes a subtraction between the observed and the expected genetic variation. So again, observed is basically the sum of pairwise comparisons, or the average pairwise comparison in your population, and expected genetic variation is this expected genetic variation given a certain number of individuals present in this population. And expected means expected if there is equilibrium, if there is nothing exciting happening. So and again, we get a negative Taji-Mazdy if we have less genetic variation than expected for the population size. So it's because it's just a subtraction. It means that if observed is lower than expected, then the subtraction of the two will give a negative value to Taji-Mazdy. Vice versa, you will get a positive Taji-Mazdy if the observed variation is stronger or higher than expected genetic variation. Again, it's all about the population size. And again, the expectation is with the larger population size, the larger genetic diversity you are expecting. Okay, and now just to clarify a bit, so as I said, you answered quite correctly on this. I just see here some few things. So some of you didn't tick this C and E. So some of you didn't tick that there is an overrepresentation of rare alleles and that the population is expanding after a bottleneck. So just to be sure, so indeed the first answer was the right one. Indeed, if Taji-Mazdy is negative, is exactly what I said before. It means that there is lower genetic diversity compared to what we expect for the size of the given population. And you all answered D correctly as well, which is basically if there is lower genetic diversity, that means that most of the individuals share the same haplotype. So there is a dominating major haplotype in the population. C is directly related to that. So by rare alleles, what we mean by rare alleles is an allele which is present in very few individuals in the whole population. So maybe 1 in 100 individual will have a specific allele. And that's directly related to this one because genetic diversity is quite low, that means that most individuals share the same sequence, and the ones that don't share the same sequence, that are a bit different, are extremely rare. So there is most like say a pool of individuals with the same genetic background, and then very few differing, and they don't differ a lot actually, they are very similar still than the major haplotype, but are still a bit different. And these individuals differing or being different are rare, so which gives them rare alleles. Okay, and because of that, there is way more rare alleles than we expect. So again, because there is low genetic diversity, it means that very few variation that exist is found in very rare occasions, in very rare individuals. Okay, and then the last one, E. So indeed it's true, so when you see a population which is expanding after a bottleneck, that is translated by having a Tadjimaz D which is negative. And why? Again, because during a bottleneck, what you're going to have is reduction of genetic diversity. And then after this bottleneck is finished, let's say the population will start to recover. So that means that you will have quite a large population size already, because the individuals start to reproduce and the population grows again, but the genetic diversity didn't recover yet simply. So what happens is you have the same genetic diversity as after a bottleneck, but with way more individuals. And that means that basically the observed genetic diversity for this already large population is lower than what you would expect in equilibrium. So because of that, then you have lower genetic diversity than expected for the size of the population, so you have Tadjimaz D which is negative. Because again, the population is growing, but the genetic diversity is not very high yet. It's really similar to the same situation as what was in the bottleneck. Okay, now so something else that was for the third chapter of the lecture. So you all seems understood quite well the point of, or let's say the characteristic of locus or gene or whatever which is under positive selection. So indeed, because, so B was correct here, because we expect that an allele which has advantageous effect will spread in the population thanks to selection. So it means that at the specific locus we will only find this specific allele. So which basically translated to having a low genetic variation, and yet we'll have a very high genetic divergence between this population and whatever sister lineage you are studying. That's of course assuming that the positive selection occurs only in this population. So because it occurs only in this population, you will have a specific allele found there which spread and conquered, let's say, the whole population, but this did not happen in the sister lineage. So low genetic variation within this population, but very high genetic divergence between this population and sister lineage. So low genetic variation compared to what is expected at the specific locus means low Tadjimaz D again, and we always need, remember, to compare this to the rest of the genome. If you only have the Tadjimaz D value for this gene only, you will never be able to say if it's because of demographic events. So low Tadjimaz D then would mean population expanding after a bottleneck, or if it's really because this specific gene is under positive selection. And only comparing this, the Tadjimaz D of this gene versus the rest of the genome, then you will be able to tell something about it. And last is exactly the same idea. F, same idea as B, precisely is what FST is measuring. It's comparing the intra-population variation versus the divergence. And if the divergence is much higher than the intra-population variation, then you will have high FST. So basically, B here is exactly measured by FST. So you will have high FST between the population and your sister lineage for this specific gene. And again, of course, this is comparing to the rest of the genome, otherwise it doesn't make sense. Or let's say that here, FST is also used to measure divergence between lineages. So if you only have the value of the gene, that doesn't tell you anything. But okay, but there was a bit more problems with purifying selection, as I could see. Just a reminder of what purifying selection or negative selection is about. So purifying selection is selection that will act against any new mutation. So any new mutation will be purged, and it will be purged because the new mutation will be deleterious. So basically, that we can also say it like that, that we will have a conservation of the ancestral state. Again, why? Because any new or derived mutation will be discarded in the population. So we will have intralineage low variability. And because, as I said, we conserve the ancestral state, we also expect reduced divergence between two lineages, because the ancestral state will be here and conserved in the two lineages. A good example of genes that are under purifying selection is what are called these core eukaryotic genes. So these genes that are very conserved between all eukaryotes. And why they are conserved? Most probably because they are under purifying selection. And why they are under purifying selection is because these genes encode a very basic and essential function for eukaryotic cells, such as the membrane, or the nuclei structure, or the cytoskeleton, like microtubules, this kind of thing. So because of that, these genes cannot be modified. If they modify, or let's say this protein their encoding is modified, it will have drastic changes on the overall cell structure itself, which means very deleterious consequences. If microtubules are disrupted or have different shapes or different affinities for molecules, the whole cell will basically be destroyed. So these genes are under very strong, what we call genetic constraints. They just simply cannot evolve anymore because they are sort of trapped. Their function is sort of trapped, and are so again so needed for the overall general functioning of the cell that they are under strong purifying selection. And because they are that, then it means that they are very conserved between all eukaryotes. And again, it's exactly what is here. Okay, and now I ask you this. So let's imagine that we have these three possible scenarios. Either a locus is under neutral, under positive, or under purifying selection. And this is a case of human versus chimpanzees, as you saw before. And remember, so red is color red is mean derived, a new mutation in the lineage. So here means new mutation in human lineage, and white means ancestral state. So something which was already present in both lineages before they diverged. So I ask you here to find sites under positive selection in humans to start with. So again, press pause and find sites under positive selection. And remember, again, no, I don't tell you anything. So just press pause, think about how we detect locus under positive selection, and find them, and then we start again. Okay, so these are three sites potentially under positive selection, and why we can detect that is because the diversity within humans is extremely low, which suggests that the allele or the derived mutation here has spread across the lineage, which indicates a positive or advantageous effect of this allele. That's the one that then went on positive selection. And what makes us tell that is because if you compare the diversity within humans, which is extremely low, with the divergence between human and chimpanzee, you see that divergence is very high. So you don't find this mutation at all in chimpanzees, suggesting indeed that diversity, what you have here, the ratio of diversity extremely low with divergence extremely high, gives a very negative value. This is log transformed anyway. So diversity very low, divergence very high, and FST shows exactly the same idea, except it's opposite. FST is rather, the opposite. It's rather the ratio of divergence versus diversity. So very high divergence, very low diversity gives you very high FST at this specific loci. So here, here, and here. Okay, so next Now I'm asking you to find the site or several sites that are potentially under neutral selection. So think about what a site under neutral selection is, and then we can continue. So a site under neutral selection means that a new mutation arising there in a specific site will not have any impact on the fitness. That's the case of most derived mutations, and we see that most of the genetic diversity is driven by demography, stochastic events, and not by selection. So neutral selection means basically no selection on this specific mutation. And of course, if there are two, let's say, kinds, if we talk about genes, a mutation can affect the protein sequence or not. If it doesn't affect the protein sequence, it will be mostly under neutral selection, and if it affects the protein sequence the gene encodes for, then the likelihood is that most of these, what we call non-synonymous mutation, will be deleterious. Okay, but most of the time a mutation is completely silent regarding selection, which means neutral selection. So the scenarios that could fit such idea is this one here, for example, where you can have accumulation of derived mutations here, there, and here, and also in chimpanzee here, which seems relatively random. The accumulation and the spread of this derived mutation in the population seems to happen in a relatively random manner. So there doesn't seem to be any gain selection acting on it. So this is here, here, here. And the way that, because of course this is relatively arbitrary, to say just, okay, it seems, the mutation seems to spread in the population in a relatively random manner, then again we use a way to measure this. And the way we use to measure this is, for example, with FST again. And we can conclude that there is neutral selection if diversity within a lineage will be relatively equal to the divergence between lineage. So for example, if we take this case, there is some diversity because half of the individuals in the human share this derived mutation. So there is some diversity, but on the other hand, there is not much divergence. As you can see, why? Because the mutation, the derived mutation, is actually shared between human and chimpanzees. So the divergence here will be relatively low, and which overall gives you a relatively low FST. That will basically be the background signal of FST, which is neutral evolution. Okay. So again, neutral selection means, well, it means basically no selection acting, and the mutations, because they don't have any impact on the fitness, will just accumulate according to stochasticity and or demographic events, and nothing else. Okay, and now what about this one? So think about it, and what do you think this site is under? Which kind of selection this site is under? So here what we have is the last case of selection. We have the case of purifying selection. Again, purifying selection is selection against any new mutation arising, so the ancestral state will be conserved. And it's exactly what you see here. You see virtually no variation whatsoever within human species. And more precisely, it's always the ancestral state which is there. So that's strongly suggests that there is, again, selection against any new mutation rising. And not only that, you see that this region is very well conserved between human and chimpanzee, which also has the ancestral state, so that all of it suggests that this locus is under strong purifying selection. So conservation of the ancestral state, and absence of diversity whatsoever within the species. And if we think in terms of FST, well, that's gonna be harder to detect simply because FST again makes a ratio between divergence and diversity, and because divergence is close to zero, because there is completely sharing of alleles between human and chimpanzees, and because diversity is also very low, there is absolutely new mutations being present, then low as a ratio of something else which is low, it's gonna be something close to 1, so something close to the background in general. So similar footprint as neutral selection. So then gives back to the quiz. So if you would answer the quiz again, you can do it and press pause again. So how can we see that we have purifying selection in the population or in general? So press pause, think about it again, and let's see if you get a better result this time. So actually the only answer that is correct is C. So if you have low Tagmas-D compared to the rest of the genome, it again suggests that at the specific locus under purifying selection, you have reduced genetic diversity compared to what you expect, which is exactly what purifying selection is doing. Again, it's purging any new mutation arising, so it's going to deplete completely genetic diversity. So it's a low Tagmas-D that we expect here. Here, none of them are correct. None of them are correct. Why? Because what we expect with purifying selection is low genetic variation and low genetic divergence. So low genetic variation within this population and low genetic divergence between the population and the outgroup lineage. And same here, basically, we're probably not going to be able to detect purifying selection just using FST. Simply because, as I said, we're going to have a ratio of low divergence versus low diversity, which ends up as a sort of neutral FST, similar to a neutral selection scenario. So that's it. I hope it's clearer now. If it's not, again, feel free to comment or whatever, and I can try to be clearer. So, cool. Anyway, well done.